Infliximab is the name of my protein. It's also known as Remicade. What's interesting about infliximab is that it's a human-mouse chimeric IgG1 monoclonal antibody that is being used as a drug to treat inflammatory bowel disease, IBD. IBD involved chronic inflammation of the digestive tract, for example, Crohn's disease and ulcerative colitis. In addition, infliximab also treats rheumatoid arthritis. But what is chimeric monoclonal antibody? It's an antibody made by fusing the mouse antigen binding region with the human constant region, Fc. The antibody is about 65% human. The benefit of using the chimeric antibody is that it's because it's cheaper than the humanized antibody. It also reduces the risk of a reaction to foreign antibodies from non-human. Here's the picture of it. It consists of beta sheets and loops. The antibody has a FAB domain, it's the antigen binding site, and a constant region Fc. So in chimeric antibody, the Fc domain is from a human antibody, and the FAB domain would be from a mouse antibody. Here's a simple version of the chimeric antibody. But how do you make it? First, we have to transfer the human immunoglobulin genes into the mouse genome. Then the transgenic mouse is vaccinated against the desired antigen. This process is called immunization. This will generate antibody-producing B cells. Then we have to determine if the mouse is producing the antibodies of interest by performing test bleeds. If the desired antibody is produced, then the spleen is removed and dissociated in the culture medium. When polyethylene glycol is added to the mixture, the B cells will fuse with the myeloma cells to form hybridoma cells. The hybridoma cells can then be separated in het culture medium. Then we culture the cells individually. The next step is screening. The last step is to grill the desired antibody in mass culture, and that's how you get your chimeric antibody. So how does infliximab treat inflammation? In order to understand its mechanism, we first have to look at the cause of the inflammation. The immune system does not respond to the body cells, which in people with inflammatory bowel disease, and that leads to an overreactive immune system and chronic inflammation in the GI tract. One of the major signals of inflammation is a cytokine known as tumor necrosis factor alpha, or TNF alpha, which is produced by some cells of the immune system. When TNF alpha is released from cells, it interacts with other cells through the TNF receptors on the surface of the cells, and this leads to inflammation. Inflammation leads to ulcers in the intestines. So how does infliximab help reduce inflammation? Infliximab is the anti-TNF alpha agent. It has a high affinity binding to TNF alpha. It works by attaching to free-floating TNF alpha outside of cells as well as transmembrane TNF alpha found on the cell surface. Since it is attached to infliximab, the TNF alpha is unable to interact with its receptor on the cell surface to cause inflammation. And this is the way infliximab helps reduce inflammation. The TNF alpha exists in monomeric and trimeric forms. This is the monomeric of it. Here's the picture of the infliximab TNF alpha complex, where the infliximab binds to the trimeric form of the TNF alpha. The center is the trimeric TNF alpha. This is the FAB domain of the infliximab. This is also the top view of the complex. Here is the side view of the complex. So let's take a closer look at the interaction between the infliximab and the TNF alpha. Here you can see on the left is the infliximab FAB domain by itself, and this is where it interacts with the trimeric form of the TNF alpha. The bottom pictures are more close-up pictures, and the blue and red areas are where the interaction happens. Now let's go back to the TNF alpha structure. These are the E and F loops of the TNF. These loops are very important and they play a crucial role in the antigen-antibody interaction. According to an experiment done at the International Joint Cancer Institute in China, the results showed that a mutation that happened in these loops results in an increase in the KD value by 1,000 times. So this mutation decreased the binding affinity, so therefore they play a crucial role in the interaction of the TNF with the infliximab FAP. In addition, a mutation in the arginine residue 138 and tyrosine 141 would result in the decrease in the binding of the complex. So because of its high affinity to the TNF alpha, infliximab can reduce inflammation. Therefore, infliximab can be used to help treat Crohn's disease and ulcerative colitis. This is the end of my video. Here are the references.